So today I'm going to talk about some of our recent work in bridging multiple simulation techniques in order to bridge scales in kinetic simulations. This way? Oh, I'm sorry. Okay. So whole cell modeling is a grand challenge for computational biology. Modeling whole cells using atomistic potential-based methods is not really feasible, unfortunately, especially over cell cycle timescales. We're lucky, though, that chemical reactions alone can account for a lot of the phenomena driving living cells. So if we focus our attention to chemical reaction dynamics, we can actually learn a lot about life in that sense. Biological kinetics are biochemical kinetics can be thought of as split on two different axes. First axis is how fast particles diffuse within the simulation volume, and the other axis is how concentrated they are. I actually think I have these. So, yeah, I got these backwards. Sorry. But the basic idea is, for chemical kinetics, you typically think of them in terms of ordinary rate equations, like you've seen in your undergraduate chemistry courses. Once you start bringing in particles which have varying diffusion coefficients, then you have to think of a spatially resolved sort of representation. And these are typically treated with partial differential equations using the reaction-diffusion equations. On the other side, when you have small particle counts, you have to start worrying about fluctuations. So for quantities which are the size of a molar concentration, the fluctuations in concentration is usually on the order of 1 over the square root of the number of particles. But when you get to living cells, you have tens, you have thousands of particles. Those fluctuations become important. Even worse, in living cells, you have spatially varying diffusion coefficients based on the different sorts of organelles in the cell. So you have to treat the spatial dependence as well as the fluctuations. And that makes things difficult computationally speaking. So I'm going to skip over these, excuse me. Okay, so what I'm going to talk about first is our coupling of the well-mixed stochastic methodology using the chemical master equation with ordinary rate equations. And our model system that we're going to look at is just simple yeast. To give you an idea of what the different scales are, cell is egg-shaped. It's about 4 microns in diameter. And for a genetic switch, we're going to consider sugar, which is in the environment with a concentration of, like, say, 1 millimolar. Sugar has a really high diffusion coefficient, especially or even in cytoplasm. It's approximately 200 microns squared per second, which means it can be considered to be well mixed in milliseconds. Protein, on the other hand, can be found in micromolar concentrations, so thousands of particles in a cell. And it has a diffusion coefficient that's 40 times slower than sugar. So in those circumstances, we do need to account for spatial dependence. And finally, there's also genes in the cell. And genes are usually found 1 or 2, maybe more, depending on the particular organism. And those don't typically diffuse, but they do move. So what you can see here is there's a separation of concentration scales, a pretty wide separation. And we're going to take advantage of that in order to speed up these simulations. So a quick little description of what how the galactose switch works. I don't really want to get into the really nitty-gritty details, but the basic idea is there are genes which code for transcription factors, for other proteins which interact with those transcription factors, and transporters. So G2 is a transporter which is found in the membrane, and it transports galactose into the cell. So when galactose enters the cell, it starts to interact with these G3 species here. So this green blob is a repressor which is normally bound to the DNA, but it can fluctuate on and off, and it can also enter and leave the nucleus. But when this G3 species with a sugar bound to it interacts with the repressor, it can no longer re-enter the nucleus, so it's sequestered in the cytoplasm. So normally the cell expresses a couple hundred or maybe 1,000 of these G2 transporters, even when there's no galactose in the environment. So when you shock the cell with a large amount of galactose, even though you have relatively few transporters, you still see a large number of reactions per second. And what that means is if you're trying to simulate this using a chemical master equation type approach, you're spending the vast majority of your time dealing with the transport of galactose into the cell, and that's a waste of resources. On the other hand, though, the switch is fundamentally stochastic. The induction of the switch is a single molecule event. It's the debinding of the repressor from the DNA. So we have to treat both ends. So the technique that we use is just a simple split of the system into a deterministic and into a stochastic regime. For the stochastic regime, we treat all of the protein species, the majority of the reactions in the system, using the chemical master equation. Specifically, we model them with the Gillespie algorithm. For the galactose, we treat those using just standard rate equations, but we also account for the transitions between different transporter states and also the decay of galactose by the metabolic enzymes that actually utilizes the sugar. So those two regimes are coupled with a fairly simple sort of method here. We simply just advance time from the CME solution up to some communication time, which I'm going to call tau. We then communicate the state to the ODE solver, and we then propagate the ODE solver again by tau and communicate the concentrations back. So it's a pretty simple scheme, but it works remarkably well. We end up with about a 40 times speed up with very little impact on accuracy. So the wall time to simulate, the wall time to simulate 150 minutes of the switch, which includes the time for it to induce and a little bit of time for it to reach steady state. It's about 47 hours on blue waters for a single trajectory. And then we need 1,000 of those trajectories. This is at high galactose concentration. So 1,000 trajectories, 47 hours, it's a lot of wasted cycles actually. But if we do this coupling, we can get about a 40 times speed up with a pretty long-term communication time, and using a much shorter communication time, we still get about a 20 times speed up. So the distribution of transporter counts at steady state pretty much all falls on top of each other for the different communication times and the pure CME simulations. Concentration, or the count is a function of time, the mean count, very similar. The time that it takes for the switch to reach 80% induction all falls on top of each other. So it looks very promising. The communication time, the choice of the communication time appears to be important though. If you get too short, if you take too short of a communication time, the G2 copy number distributions between the CME and the hybrid simulations start to diverge, and there's probably some sort of instability there that we're not really understanding completely well. And of course, at larger, much larger communication times, it's not enough time to communicate the change in the system fast enough between the different solvers. So we found the sweet spot was about 100 milliseconds or so. So that was pretty, we were happy with those results. So we then took those results and tried to apply it to a spatially resolved system. So I don't really expect people to be super familiar with the reaction-diffusion master equation. So I'm going to give you kind of a quick little explanation of what it's about. The idea is we're going to segment the environment into a bunch of different region types. So you can imagine that this is an E. coli cell that I've cut part of the side off and all of the bottom off. This is outside the cell, this is the cell membrane, this is the cytoplasm, and this is the nucleoid region where you would find DNA. So throughout the environment, there's going to be a bunch of different chemical species. This could be sugar, this could be a gene, etc. So the idea of the reaction-diffusion master equation approach is to discretize the entire system onto a lattice. Each subvolume is then is its own little well-stirred reactor, like in terms of the chemical master equation. So each one of these subvolumes has a site type associated with it. So this is extracellular, this is cytoplasm. And as well as the site type, it has the count of every particle type in the system. So this has one black, this has one black, one green, 0 red, 0 yellow, etc. So particles can then react with each other within subvolumes, or they can diffuse between subvolumes. And that's the extent of the dynamics in the RDME methodology. So the entire system state is described by the particle count at each subvolume. That's it. That's the entire system state. So we have been working on software to simulate the reaction-diffusion master equation for a number of years now. It was first written by Elijah Roberts back in 2009, and we were really lucky because that's when CUDA was really starting to become a big deal. So we were lucky enough to jump on the bandwagon. We recognized how great of a problem that the RDME solver is for application of CUDA. So we immediately got out a publication involving diffusion in 2009. Then our research programmer, Mike Halleck, came out with a multi-GPU version, which does dynamic load balancing across multiple GPUs on a node. And we are currently working on an MPI version. It's pretty close to done, but it needs some further validation. So our solver is extensible using Python. So you can just load up a Jupyter notebook, design your simulation in Jupyter, and then run the simulation all on the same notebook. We took advantage of that extensibility so that we could actually write these hybrid solvers. So for the yeast switch, we need some sort of geometry in which to run these reactions in. So the geometry that I chose to use was from cryo-electron tomograms of yeast from Elizabeth Vila's lab. So basically what we have here is a 2 micron by 2 micron by a 1/4 micron segment of a yeast cell, and this is intersecting part of the nucleus and part of the cell wall. This is the cell wall out here. And this only takes up 2% of the volume of the cell, so we can't actually use this entire thing to run our simulation in. So instead what we do is we use it as a template to build the whole cell geometry. So we're going to build an idealized cell using this. So starting from a cross-section of the cell, we can see the nuclear envelope, we can see the cell membrane and the cell wall. There's ribosomes out here. Starting from the segmentation data, we can then fit an ellipsoid to the nuclear membrane to get the size of the membrane. And then counting the number of holes in this little segment down here, we can get the density of nuclear pores and put those into the membrane. So this is all we have of the cell membrane. So we can't really expect a fit to that to do anything really, really valid. But what we can do is we can use a fit to the cell membrane little piece down there to position the nucleus within the cell volume. So we fix the volume of the cell and just fit an ellipsoid to that. And finally, once we have all of this stuff fit the way we want it, we can add a bunch of idealized organelles such as ellipsoids for mitochondria and say a vacuole here. So the nice thing about these reaction-diffusion master equation simulations is they're very visual. So what I'm showing here is the switch, the induction of the switch. We start off with a gene in the, oops, excuse me. I hit the button. Okay. So we start off with a gene in the middle of the nucleus in the off state. So we have a basal amount of transporter on the membrane. Those are those little flecks here. They're cyan-colored flecks. And we have 0 of the galactose inside the cell. So as galactose starts to come into the cell, we start seeing more and more of these transporters produced. Well, actually, we don't see the transporters being produced yet. We have to watch the state of the gene. It turned green, which means it switched on. And you can see these little diffusing spheres here are mRNA, which are interacting with ribosomes to form more of the G2 protein. So you can see that the G2 count reached kind of an inflection point here. So the gene is induced, or the system is induced at this point. And you can see that the density of transporters is increasing a lot. So the resulting density is kind of asymmetric, which we found to be somewhat interesting. It's not especially surprising since all of the mRNA are being produced here, which is near this part of the membrane. So the nice thing about these RDME simulations is we no longer need to have different types of chemical species to represent stuff that's in the nucleus and stuff that's outside of the nucleus. We have one species type and it comes in and it comes out of the nucleus, and we leave it at that. Which means that we can see that the G80 is actually sequestered from the nucleus without having to do any sort of juggling with the rate equations. I mentioned that there was some sort of weird distribution of transporters, so I took the density of transporters in the membrane and then projected a polar projection of that. And yeah, it looks like there is some sort of higher density in this region here, but I think that's probably just an artifact of the way the simulation was designed. I would not expect to see this in a real cell just because there's the endoplasmic reticula, there's other organelles which help bring proteins to where they should be inside the cell. Okay, so we've been able to demonstrate the applicability of these rate equation hybrid models with the well-stirred situation and the reaction-diffusion master equation. We're planning on doing a PDE model in the future, but we haven't gotten there yet. And currently, I am working on a Brownian dynamics-based hybrid model, which will account for large particles interacting with a lattice spacing, which is much smaller than the particle size. So the whole goal of this hybrid modeling work is we want to simulate an entire cell. So that's kind of a lofty goal. Fortunately, there is a cell which is just simple enough that it's probably possible. It's the so-called minimal cell. The Craig Venter Institute has created this synthetic cell based on Mycoplasma mycoides, and they systematically removed genes from its genome until they were left with about 450 some odd protein coding genes. But at the same time, it's still able to grow robustly in a rich media with a doubling time of 2 hours, which makes it a really useful experimental type model while being incredibly simple at the same time. So this figure here shows a comparison of the two genomes between Syn3A, the minimal cell, and E. coli. These are Voronoi tree maps, so each little cell, and the tiniest cells represent single genes. The colors represent different functional classes. These are metabolic genes. These are genes that are responsible for gene expression, replication of DNA, etc. And these are genes that have unknown function. In the minimal cell, we have 91 genes that we have no idea what they do. That's really impressive in my opinion. Also, the size of the genome is tiny compared to E. coli. There's about more than 4,000 genes in E. coli. There's 450 in Syn3A. Because of this, I think it's reasonable that we could come up with a whole cell model of the minimal cell that covers the entire genome. And to get there, we have to use hybrid methods such as ODE modeling of metabolism, RDME for gene expression, and Brownian dynamics to model the larger objects such as ribosomes, DNA, and the cytoskeleton. So my advisor is Zan Schulten, and David Bianchi worked on the CME ODE work. Mike Halleck is our research programmer who does a lot of awesome things, and Joe Peterson was also involved in the CME and ODE work. Thank you.